Hi, I'm Harris and I'm a new PhD student working in the Striker group. One of the main aims of our group's research is to understand the epidemiology of viruses and to find ways that we can preempt these viruses from emerging in humans. I think the COVID-19 pandemic has opened a lot of people's eyes to the devastating impacts that can happen when viruses spill over into human populations, particularly from wildlife. In Latin America, where most of our work takes place, rabies is still a huge problem. In these countries, vampire bats are the primary source of rabies, and spillover from these bats results in huge losses both to human lives and in the livestock industry. Rabies has been classically controlled in Europe and North America through the vaccination of animal reservoirs such as dogs and foxes. However, for vampire bats, these approaches are currently not applicable, as bats are naturally elusive and their colonies are hard to access. Furthermore, current control measures such as culling and livestock vaccination have proven largely ineffective. One potential solution here is the use of self-spreading vaccines, whereby harmless vaccines are modified to express a protein from a target virus which would elicit an immune response in the target animal, whilst retaining transmission capacity to spread to other animals in the population. This would allow, theoretically, vaccination of a larger bat population by only directly interacting with a few of them. Using this novel technology, we could potentially preempt viral emergence in humans by eliminating viruses from their animal sources, in particular from animals that have been historically hard to vaccinate by hand, like bats. Previous work has found a naturally occurring herpes virus in vampire bats that shows many characteristics of an ideal transmissible vaccine. My work aims to develop a transmissible rabies vaccine by isolating this virus and modifying it to express the rabies glycoprotein. Following this, I will undertake experimental infections with this vaccine in captive vampire bats to determine critical properties that would underpin the vaccine's potential success and deployment, such as its ability to infect bats, the strength of the immune response generated, and the transmission capacity. This is obviously quite an ambitious project, but with success, the implications could be huge. Locally, we'd be significantly reducing the rabies burden in Latin America. However, globally, we'd be providing a framework for using transmissible vaccines to control other significant zoonotic viruses.